Hello, everybody. My name is Roman Bauer, and I would like to talk about learning how to improve the diagnosis of and think about human diseases with computational models. First of all, I would like to highlight that computational methods are becoming more and more used for biomedical applications, such as, for example, the diagnosis of and monitoring of medical conditions. And one of the main reasons for this is that there are increasing amounts of data available, acquired from different kinds of technologies and equipments, which can be very complex. And so computational methods are very well suited to address these challenges. And relevant problems for such computational approaches include conditions such as Alzheimer's disease, Parkinson's disease, cancer, cardiovascular disease, and many others. Now, I would like to focus on here on the brain. And the brain, as you know, is a very complex organ. It's actually the most complex entity known to mankind. And here you can see a visualization in 3D. This visualization was obtained based on data from diffusion tensor imaging, or DTI, which is a technique that allows to measure the diffusion of water molecules. And by doing those measurements, one can actually infer fiber tracts and connections between individual regions or areas of the brain. And here you see in different colors, you can see different kinds of fibers and their orientations. So red would be horizontal orientations and blue vertical or approximately vertical orientations. And you can clearly see that the structure of this human brain is indeed very, very complex. And it is indeed too complex for a human to see striking differences between different conditions of those brain structures. And so one thing, one example that we have done in the past using such diffusion tensor imaging data is here's a schematic of such a piece of work that I have done myself with my colleagues in the past. We have used connectivity matrices. So these are data structures that make use of this DTI data and essentially create an abstract representation of the brain structure in terms of brain connectivity. And what we did is we used data from the database ADNI, which is a very well-established database for researchers who are interested in Alzheimer's disease, in order to essentially employ machine learning and see whether we can differentiate those healthy subjects, so control subjects and subjects with Alzheimer's disease. And indeed, we show in this publication that we can actually diagnose or classify different types of brain connectivity networks based on whether they're healthy or have Alzheimer's disease quite early on, actually. So what we did is we simulated the progression of aging in normal aging and in aging of subjects with Alzheimer's disease. And we included a type of staging process, so we essentially created a way to quantify the stage of this progression. And it turns out that we can, with high accuracy, classify healthy versus diseased brain networks earlier than what is currently possible in clinical practice. So on the x-axis here, you see the different stages. And as you can see here, that the classification performance increases consistently with the stages. And then at around stage -4 or -3, you get a very, very high classification performance. And you can also see this statistically by looking at the p-value, for instance. where again with progressive stages, you can see that the p-values reduce very significantly, and you can actually think of a stage approximately as indicating a time span of 1 year. So based on these results, we have good reason to believe that this method that takes into account DTI data and structural data, not functional data of the brain, we can diagnose at least the risk of having Alzheimer's disease earlier than what is currently possible. And so this is one example where we have used machine learning, and these are actually quite well-established machine learning approaches. So they are not very novel machine learning methods, but they have existed for many years in computer science. And so this shows that by using such well-established methods, we can actually apply them to a biomedical condition, in this case Alzheimer's disease, and use them to make certain classifications that have applicability in a clinical setting. And as I said, this was now by using structural data, but there is also a lot of other ways of how one can obtain information from the brain. It doesn't have to be only on the structure or on the fiber tracts between brain regions. Another thing that we're actually currently also pursuing in more detail is we have looked at so-called retinal fundus images. So retinal fundus images are images taken from the rear of the eye, where there is the retina, which is a neural tissue, which is actually part of the brain, and we use machine learning methods, in this case so-called convolutional neural networks, so CNNs, in order to infer certain features from those retinal images, and we can then classify, for example, different blood vessels according to whether they are arteries or veins. And then we can take this further and look at the structure of this. And we show in this paper here that, again, we can use machine learning in order to classify subjects' retinas, depending on whether they're healthy or whether they have Alzheimer's disease or Parkinson's disease. And so if you're interested, I would recommend you to have a look at this publication. But essentially, this is just another example of where we have used machine learning methods in order to apply to a medically relevant topic. And one of the very nice aspects of this work is that retinal images are very easy to obtain. So essentially, one doesn't need a lot of expertise, and it's a very efficient and also associated with low cost method. And just to conclude this part on dementia or Alzheimer's disease, we have also looked at so-called EEG data, so electroencephalography, where we looked at different subjects. Some of them were healthy, some of them had certain types of dementia. And again, we could differentiate using machine learning very well and with high accuracy whether those subjects were healthy or had some dementia types diagnosed previously. So of course, this previous diagnosis was done independently of the EEG, and we could show that we could very well reproduce those diagnoses. Here you see the accuracies, which are very high for Alzheimer's disease, dementia with Lewy body, or Parkinson's disease dementia, which are sometimes in practice sometimes very difficult to differentiate. And this then also has implications with regards to treatment. So so I would like to now conclude this part on using machine learning for dementia subtypes. Another approach, which also is very promising and that we have followed also in the past with my colleagues, is to model computationally the brain tissue in a mechanistic manner. So before, what I showed you, we used many statistical methods in order to create models that then allow to differentiate between different conditions. But what we did here in this paper, we created a computational model that can explain certain brain structural characteristics, in particular the layered structure of cortex. So the cortex is the outermost part of the brain, and it is responsible for higher cognitive functions such as planning and higher order thought processes and so on. And we used agent-based modeling, which is this mechanistic computational approach, in order to simulate how this layered architecture in the cortex develops. And then we also compared with experimental data. And one of the clinically relevant parts of this work was that we could actually put this in correspondence with experimental information from certain medical conditions, including polymicrogyria. So in polymicrogyria, the cortex is much stronger folded than in healthy control conditions, and we could actually reproduce some aspects of the cortical structure in polymicrogyria using such mechanistic computation modeling, which then, of course, in the future could be elaborated on in order to better understand mechanistically what exactly are the genetic changes that are necessary in order to produce this altered brain structure. And then ultimately, also, of course, the aim would be to find potential interventions that can mediate or cure such diseases of the brain. Another aspect that we looked at is autism spectrum disorders, and one of the features of autism spectrum disorders is that there is some different arrangement, spatial arrangement of cell types. You can see here that this spatial arrangement here appears less ordered than here. And again, we could use computational modeling and simulation with agent-based modeling that actually reproduced such differences or characteristics of these. And again, of course, this better computation models can also help us better understand and gain insights into how those conditions develop over time. And of course, these kind of models often require a lot of computational resources. So they need a lot of computer hardware and very cutting edge and performing hardware. And in order to address this challenge, we created a software called Biodynamo. So the Biodynamo Consortium, which I'm the spokesperson of, has created this software, Biodynamo, and we published this recently. And we present essentially the software, and we have shown that we can now simulate with very high performance, very complex systems and large systems of biological tissues, including neural tissues. And importantly, we provide this code or this software as open source for the community. So if you're interested in this kind of agent-based models where you have mechanistic models of how tissues develop over time, where you can model this, or other systems, of course, then please have a look at this website. And there is also installation guides and tutorials and example demos and so on. And of course, feel free to reach out to us either via the website or directly to me. Importantly, we also use Biodynamo not only for research, but also for didactic purposes. So we have, a few years ago, we have engaged with Intel, and we have attracted almost 20,000 students from all over the world in a competition, which helped many students learn about computational biology topics, but also then it actually gave rise to a publication. And another example, so in this case, we are not looking at Alzheimer's disease. We have here also simulated glioblastoma. So this is a type of brain tumor where we simulated using biodynamo how the glioblastoma progresses over time. So how does the structure of glioblastoma change over time, and how do different aspects of glioblastoma change over time? So for example, we look at oxygen saturation, we look at density of certain types of cells, because these are very heterogeneous cancers. And so creating such models, computational models, can help us better understand how the cancers might progress over time within weeks or months, and also ultimately then help advise or help with coming up with treatment options. Like they can help ultimately with optimizing treatment parameters, such as, for example, what type of drugs may be expected to have the highest impact, or at what concentrations, and so on. Because currently this is an area where doctors' expertise is used, of course, and it will be used also in the future. But by using computational tools, one can improve essentially the accuracy and make this more objective, so that we don't have very different recommendations from different doctors, but we can create like objective models that allow to in interaction, of course, with medical doctors, to better pinpoint what kind of treatment strategies may be the best ones. And just another example, again, from cancer, would be about fibrosis of the lung. Very recently, we published this paper here, where we could reproduce certain behaviors that are well established in terms of the relationships between the dosage of radiotherapy and changes in the extracellular matrix concentration. So here you can see this good agreement with experimental data. And again, this is just another example for how computer modeling, especially with regards to the mechanistic approach, so agent-based approach, where we really model individual cells and their interactions and their individual dynamics, how this can be used for medically relevant applications. In this case, it could be used potentially in future to optimize and reduce fibrosis as much as possible, because this is a big side effect from radiotherapy of the lung. So to conclude, I would like to highlight that computational analysis and modeling is becoming more and more important in biomedical applications, and that there are certain data-driven methods, like machine learning methods, that create statistical models that allow, for example, to diagnose certain pathological conditions such as Alzheimer's disease. But then there is another alternative approach, which is mechanistic modeling using agent-based modeling, such as, for example, biodynamo, this open source software. And this can then be also used directly to test different treatment scenarios, which could then help medical doctors in coming up with treatment options. So, of course, there is a lot of future research that needs to be done. We need to better understand those diseases, how they progress. We need to take into account various types of data and various amounts of data in order to iteratively refine the computational modeling and make them also then more and more relevant. For example, also in a personalized medicine approach, so in silico medicine. So finally, I would like to thank you for your attention, and I would like to thank my colleagues. I've listed some of them here, but I cannot list all of them who contributed so much to all this work that I've shown you now, or briefly given an overview of. I would like to thank, of course, also my lab, who are very prolific and very interactive. And I would like to thank the funders who have made it possible to do this research. And if you have any questions, please do not hesitate to get in touch. This is my email address and Twitter handle. And so thank you for your attention.